So I'm going to go ahead then and introduce you and talk a little bit about NOHBA for a second, and off we go. So thank you to everyone for joining us today. I expect to see a few more people sign in. Usually there's a little lag time. Today, Dr. Larry Weiss is sharing some of his really long time knowledge on the microbiome and on a product line that he's developed based on that knowledge. And I've known Larry for, I don't know, 10 years or something.
Wow.
Whenever you were working at Cleanwell. So I've known him through Cleanwell and then Mother Dirt. And it's been a pleasure, and it's always interesting. So NOHBA, for those of you who may not be fully informed about it and just saw it on Instagram, is the Natural and Organic Health and Beauty Alliance. And we are a small nonprofit working to create and provide resources for this clean space and trying to help facilitate the conversation about what some of these words we're using mean: clean, green. I don't think we're going to talk about conscious beauty, but any of these other terms, I think we're going to try and just really help provide greater information and greater conversation about them. But in the meantime, we do these webinars. And so Larry has been kind enough to agree to do the one he's doing today. And the next one will be on formulating with shea butter, because that's a pain. And a lot of people use it in their products. And I had somebody calling me and complaining, and I went, oh, I know somebody who knows a lot about that. So keep an eye out for that one. And I would say now, Larry, take it away.
Yeah, thanks, Gay, for inviting me to do this. First, I want to make this as much of a conversation as possible, so feel free to interrupt at any time. What I'm using here is, this is a presentation that has evolved over time, basically as a way of sort of introducing the microbiome. I can talk a little bit about the products and what it is that we've done with them, but mostly this is sort of kind of a journey through the microbiome. And like any journey, where we are today is a function of how we got and what we've learned along the way. So I'm going to go back in time and sort of talk about how we've gotten here in the microbiome, and then I'm going to challenge your notion of what you think of as health, and I'm going to invite you to consider that what we're learning in the microbiome is changing that and transforming that, and hope to invite you to participate, because whatever it is that you're doing, whatever your product is, whatever your activity is, it has an impact on health. So I think if we start rethinking our notion of health in this way, it'll benefit all of us. And then I'll talk about some of the work that we've done in the Amazon, looking at what the human microbiome looked like 12,000 years ago. And I will finish up by challenging your deeply held beliefs about what it means to be human in the world in which we live today. Okay, so let's see if I can do this. All right. So first, we chose the name symbiome because it's actually a technical term. And what it is it's all of the co-located, co-evolving life forms in any defined environment. And we can define that environment as tiny as a small part of your skin or your gut, or we could define it as the entire planet. But the big lesson in the microbiome is that everything is connected, and almost all of those connections are microbial. And we'll go into that some more. So let's go back in time to 1958. And this notion of what we've learned about the microbiome started in an offhand remark that Francis Crick of Watson Crick fame made in a particular paper. And what they were doing is they were looking at animal hemoglobin. And hemoglobin is, protein. It's a sequence of amino acids. And just in this little offhand remark here, the most delicate expression, the possible phenotype of an organism, and that vast amounts of evolutionary information may be hidden within them. He noticed that differences in the sequence could be used as essentially a barcode or a biological clock on an evolutionary scale to look at what a specific species was and where they came from. This idea was picked up actually by Linus Pauling and collaborator Emil Zuckerkandl, and they came up with this idea of as a biological clock. This was picked up by this guy, Carl Weiss, and Carl Weiss was not a microbiologist. Carl Weiss was a molecular biologist, and he realized that if we just use proteins as the biologic clock, we can only go back so far. But if we went back to the machinery that makes proteins, the ribosome, we could go back from, instead of looking hundreds of millions of years, we could go back billions of years. So the ribosome is this tiny little piece of RNA. It's actually 2 subunits of it. And this is what translates the DNA into proteins. So this goes back even further. This was really early on in the evolution of life. And so what he discovered is if you take and you sequence that, what's called the 16S, which refers to the size of it, piece of RNA, you could use that as a barcode because there were hypervariable regions. There were regions of it that never changed or it lost its function, but there were other regions where the sequence could change and the function was preserved. And if you looked at just the random mutations in there, you could actually create essentially a tree of life, this phylogenetic tree of life that's down here, and you could use it as a clock. And this was revolutionary, and he introduced this into the field of microbiology. This illustrates an important point, which is the person who's going to change the field in which you work today probably doesn't work in that field today. And when that person comes in and challenges what people understand with something that is beyond their current imagination, they are generally not well received. And that's how he earned the title Microbiology's Scarred Revolutionary. And like any revolutionary, he actually introduced an entirely new kingdom, which was the archaea, which you see down here. And it was not well received. And ultimately, this transformed everything. And he was certainly vindicated. So as we developed the technology, we could go from doing these sequencing with great deal of effort and very slowly to do it relatively quickly. We chose to study our own favorite subject, which is us, and started the Human Genome Project in 1984. And with that came this advancing technology. And when we went off to study us, what we discovered is that we're animals living in an almost exclusively microbial and ancient world. And so this is the tree of life that came out of all of this. And this is basically all the life on Earth. And everything you're looking at here is microbial, except for way down here on this little branch of Pisciconta. And on this little branch are all animals and fungi. Everything else was microbial. And everything else has been around for a long time. We think of ourselves as the pinnacle of evolution. We are not. We are the spoiled and arrogant newcomers. We are the beneficiaries of this 4 billion-year-old biologic trust fund that we depend on for our very survival and seem determined to squander. So we started the Human Microbiome Project in 2007 to start studying this as the technology kept advancing. This developed into what in 2011, when this seminal paper by Elizabeth Grice and Julius Segre came along and reported that we actually had a microbiome on our skin. Our previous notion of our relation to the microbial world was only when something bad happened. We had a germ theory of disease where there were bad actors and they made us sick and we had to kill them. This started something that I want you to start thinking of as the germ theory of health. And the germ theory of health is much more larger and much more dimensional, and it is where it's, in fact, the balance of this incredible ecosystem in which we live and we depend on for our survival. And its imbalances this that set in motion things, and it's only the last couple of steps of that we see as disease. I want you to think of health like a Jenga tower, you know, the tower of blocks. And what we've learned today is if that tower was 300 blocks tall and each one of those blocks was a unit of genetic or metabolic capacity, only one of those blocks would be human and all the rest would be microbial. And what happened is that we evolved as creatures of scarcity, as hunter-gatherers, as foragers. and we had all those blocks in there. As you pulled blocks out, as we broke connections with the microbial world in which we evolved, the tower still stands. What it's lost is resilience. What we see as disease is the last couple of blocks before the tower falls. We can't see all the missing blocks because none of them have them. None of us have them. And the hardest thing to find is something you didn't know that you lost in the first place. Okay. This evolved into the Integrative Human Microbiome Project. And suddenly there's all these companies, and we behave like neo-colonialists with new technology. Something comes along and we land on it and we rape and pillage and see if we can use this to make rich people richer, because they're not rich enough yet. And we still don't know enough yet. And a lot of these companies are out there and they've got a hypothesis, or maybe they don't. And they've got some enthusiasm and maybe some investment capital, and they're out there relating to the public that we know things that today I can tell you that we hope someday that we will know, and they're burning our credibility. And someday when we know these things, maybe no one will believe us. So this is the Gartner hype cycle. If you've never seen this before, this is how we handle new technology. We come along, we think it's going to solve everything. We get all excited. We reach the peak of inflated expectations. Then we realize it's more complicated, and we're going to have to work harder. And then we end up in the trough of disillusionment, where we really don't want to work that hard. And hopefully, if we persist into the slope of enlightenment, we end up at a point where it's productive. But I want you to start thinking of productivity in terms of outcomes and not incomes, and we'll get there. But we're in the midst of a true scientific revolution. And the life sciences today is where physics was 100 years ago. And you can imagine these Newtonians 100 years ago watching this oncoming quantum revolution, dismissing it as irrelevant or just wrong. In fact, what they were doing was not wrong. It was just a small part of a much larger, much more dimensional universe they could not even imagine. And when someone from the outside came in and stretched their imagination, they were rejected. But today we're doing systems biology. We're not doing Newtonian pharmacology anymore. We see a couple of points in a row. We have a desire to draw a line through it. There's a whole lot of points we don't know about. We're not doing the same sort of linear relationships between lock and key relationships anymore. We're now looking at things where everything is connected with everything else. So what have we learned so far? Well, the first thing is, it's still really bloody early, okay? And we don't know very much. We are really at the beginning of things in the microbiome. And I'm going to use a metaphor for this. This is the Hubble Deep Field experiment. And what they did is they picked the darkest piece of space they could possibly find, and they pointed the Hubble at it. And every time it went around, it caught a high resolution infrared image. And after over a year and over 20 hours of images, what they found in the darkest piece of space that they could possibly find, I had the order wrong, is this: 10,000 galaxies. Let me go back a step because I lost my order here. The foundational methods we use to study the microbiome are this: we swab your skin or we sample your poop, and then what we've got is a bunch of stuff. From that, and that sampling introduces bias. The way we sample it determines what we collect. From that, we have to extract the DNA, and different bacteria give up their DNA with different degrees of difficulty. So it introduces a bias. They are the gram negatives that give up their DNA easily. We sequence more of those than the gram positives and some of the other things like spores that don't give it up at all. From that, every single one of these steps introduces bias. So if you look here at B, the actual composition was a third, a third, a third, but the observed composition was nowhere near that. And the people over at NIST, and their job is to standardize our foundational methods, and make no mistake, this is our foundational method. The two of them will be given a new workflow. They have a defined composition, so they know what it is. They'll run it together. They'll both get the same answer, and they will both be wrong. We are precisely inaccurate so far. So it is still very early. And this is kind of where we are. I don't know if you can see the bottom of there because my bar is, but basically that tiny little piece is the shit that you know. This one here is the shit that we know that we don't know. And there's a vast amount out there. And if we think that as the method gets better, it's going to get more, it's going to get simpler, it will not. It's going to get more complicated. This is a transformational science. We are working at the limits of our understanding. This is my first law after 40 years in life sciences, that everything's more complicated than we think that it is. Most things are more complicated than we can imagine. And this second law applies, that when we open a can of worms, we should have a bigger can somewhere nearby. This is our big learning, and that is we have lost an enormous amount of the diversity was our evolutionary legacy. We spent 98% of our time on this planet as foragers of creatures of scarcity. Our relationship to the environment in which we lived and to each other was much more intimate than it is right now, and we were deeply colonized with these. And it's not just, let's go back. It's not just that we've lost the species, and our recent studies on the Amazon are we've lost about 80%. 80% of the bacterial taxa that we were colonized with before are gone. We've also lost 25% of their genetic and metabolic contribution. We had a layer of these on our skin, and they did all kinds of things for us, and none of us have that layer anymore. More on that later. And again, as I introduced before, there is a germ theory of health, which is much larger and much more dimensional than our older germ theory of disease. But this gives us the option to do something we could never do before. And that is the difference between treating disease and restoring health. And in treating disease, what we do is we take something that's broken and we fix it. And we frequently apply things that were never part of our native physiology in doing that. We make things in laboratories that never existed anywhere on the planet until we made them. And we apply this to this complicated system. Voltaire said that medicine was the application of compounds about which we know little to an organism about which we know nothing. And I'm not sure how much further we've gotten from that. Restoring health is identifying what we had before. And I'm going to go into why this matters in a moment. But you can't restore health unless you know what it looked like before. But this opportunity to restore health, and mostly what I'm talking about is a loss of resilience in response to stress. We live in an age of epidemic inflammatory disease. It has tripled in the past 30 years. Pediatric atopic dermatitis, eczema, has more than tripled. Now 1 in 4 children has this disease. Follow those kids for 5 years, 90% are carrying an inhaler, and they have 1 or more food allergies. That is not the childhood most of you probably remember. So here's the big question, what is health? And if I ask most people what's health, it's just, I'm not sick now, at least not that I know of. But I did some research on this, which is to say I Googled it, and here's what I came up with, that it's free from being physical or psychologically or malfunction. It's wellness. Well, okay, that's not helpful. So I went after wellness, but that didn't help either. And the FDA doesn't have a definition of health. That is not what they do. They are an organization around disease and the treatment of disease. And over my career as a physician, we have changed the way we thought about it. We've took medicine, which was a profession, we turned it into a business. We became more interested in incomes than in outcomes, and we got exactly what we asked for. What you can see here is we pay more and we live shorter, sicker lives than every other developed country in the world. Everybody's making money, but we're not living longer, healthier lives for it. This was an amazing exhibit. I hope it's still up at the British Museum, and this is called Pills of a Lifetime. And so this is the pills, the prescription medications you will take over the course of a lifetime. There's 16,000 of them. Now, it's not as bad as it looks because one side is a man and the other side is a woman, but it's actually worse than it looks because this is only the prescription medications. When you throw in the other pills and things that we take, it's 3 times this size. That's what we call health. It's good business, but I'm not sure it's good health. I want you to consider that health is perhaps more than just not being sick, but that health is our evolutionary legacy. Evolution doesn't optimize creatures that have such a burden of disease as we have. At this point, I want to introduce the term terroir, which many of you know. It's from French viticulture, and it refers to the imprint of the environment on the grapes and the plants that grow there. I want to expand this concept. It's literally the biologic imprint of all of the organisms, all the bacteria, all the microorganisms, the fungi, the viruses, everything in that environment on everything that grows in that environment. And these are ancient relationships. And they are not the relationships from which disease is born. They are actually the relationships from which health is born. And humans had one. We had a terroir. But there's very few humans left on the planet that still have this. So this was a paper that came out in 2015, and these investigators went down and spent a month living with a minimally, they actually weren't uncontacted, they were minimally impacted tribe of Yanomami hunter-gatherers living deep in the Venezuelan Amazon. And what they discovered, that they had the highest diversity of bacteria and genetic functions ever reported in humans, not by a little bit. Interestingly enough, they also had in their microbiome genes for resistance to antibiotics to which they'd never ever been exposed. Now that may sound surprising to you, but it shouldn't be, because most of our antibiotics today are directly or indirectly derived from microbiota themselves. What we did is we took the molecules that they use to maintain and regulate their complex ecosystems, and to take a line from Spinal Tap, we turned them up to 11. When we do that, it triggers all of the compensatory mechanisms that are built in these communities, and those turn on those genes, and those genes get transferred horizontally to species that never had them, and that's what we call the spread of resistance. Again, entirely predictable in retrospect. But here's the amazing thing. When you look at their skin, they have no inflammatory disease at all. They don't have acne, they don't have eczema, they don't have rosacea, they don't have psoriasis. There is no inflammatory disease within this population at all. And it's not just within the Yanomami. This has been looked at in other groups of hunter-gatherers and foragers that still live traditional lifestyle. And again, no cases of active acne, no acne at all. Acne is a disease of Western civilization. This is what our skin looks like, and I'm going to take it down because I know it's gross, but it's making the point. Because our skin is the biomarker for our systemic health. It's the one we can see. It's the one we look at in the mirror. It motivates our change. We try to do things to fix it, but we're still very early on, and we're still in the process of treating disease and not restoring health. This, I'm amazed at this study. These guys actually went down and they did non-contrast coronary artery tomography on 705 hunter-gatherers in the Bolivian Amazon. I actually have no idea how you do such a thing. But what they discovered is, and you can see, from the images here, they're kind of impacted. They have some of our clothes, and but they still live a traditional lifestyle. But you see on the graph over here, here, this is their traditional lifestyle. Is on the left, you see us, and this is coronary artery calcification score. That's us. But if you see them on the right, even in their 70s and 80s, they are better than we are in our teens. Interestingly enough, they don't have diabetes. They don't have metabolic syndrome. They don't have any inflammatory disease at all. And yet, when they measured their C-reactive protein, which we use as an indication of inflammation, 50% of them have C-reactive proteins in the area where we would diagnose these people as inflamed. And what this tells us is we don't know what a healthy human looks like, because none of us are healthy. And there might be 25 or 30,000 healthy humans left on this planet, and they're critically endangered. Again, this just transforms the data a different way, but you can see right here is everyone else is up here. These guys are down here in the bottom. And if you had to choose someone to be today, it'd probably be a Japanese man. And here's how I think we got here. For most of the time on the planet, we were optimized by the evolutionary pressures and the biologic forces in which the world in which we lived. We were foragers. We didn't starve to death, by the way. They lived quite well. But if they had food, you know what they did with it? They ate it. The idea of storing food is ridiculous. And these people, they don't have a written language. They don't count time. the way we do. In fact, they don't even count. Their numbering system only goes to 2. It's just 1, 2, and many. But hunting and gathering was burdensome, and we developed agriculture. And then we domesticated animals, and then we started living in large settlements, developed chemistry, technology, consumerism. And we moved from this Anthropocene world into a Capitalocene world where we have turned capital into a biologic force. And we took things that were rare and precious, and we made them common and cheap. And when you did that, what happened is burden of disease that never existed before started to show up. Let's see if it's going to behave. There we go. Now, this is an internet meme, and I haven't fact-checked the numbers, but it's all directionally correct. But this is what's important, is this is the perceived message. Women of reproductive age see their children living sicker childhoods than they do, and they think it's something they're doing, or they're not doing enough of, or they're doing too much of. And they're looking to make better decisions. And unfortunately, the market is responding by giving them more bullshit. And by the way, bullshit is not a probiotic, in case anyone was wondering. And it's not just my opinion that this is the case. Reviso Murray from Robert Wood Johnson came out and said, we are raising the first generation of Americans that live sicker and die younger than their parents. And if that's not concerning, it certainly should be. So why is there so much inflammation? And again, this is my Jenga model. What's happened is we pulled enough blocks out. When you pull the blocks out, the tower still stands. It just can't respond to stress without collapsing. And that's in fact what has happened. So we have run 3 expeditions to the Venezuelan Amazon to see what a healthy human looked like, because you cannot restore health if you don't know what it looked like. And you may remember there was a woman who found a medieval painting of Jesus and decided to restore it on her own, and the result was not pretty. When these people are gone and nobody believes that they're going to survive, the land upon which they live has gold and oil on it. Every time we get near them, we give them bad diseases. We don't give them any of the resources necessary to take care of those diseases. And you cannot just show up in one of these villages with an IRB and a piece of paper with some words on it, have them put a thumbprint on it, and start collecting their biologic materials. The only reason that we have been able to do this is we have a unique relationship with a young man who is half Yanomami and is also a biologist, and reached out to us with the interest of preserving his people and that means his people at both sides, the Yanomami and us. So this was our expedition. We went down to build the deepest and most comprehensive model of what the ancestral human microbiome looked like when we still carried most of the biologic optimization that was our legacy. This is David in his family's village, and everyone in these village is related by blood or marriage. They are all family. These are the only humans who know how to live sustainably on this planet. And he is perhaps the most accomplished field biologist in the world today. And here's what we learned. So these are diversity plots. And if you look at the one on the left here, on the as the chow diversity increases, that means there are more species and strains. And so it is a numerical count. Each one essentially counts for one, whether it's high abundance or low abundance. And the Shannon diversity, which is the next plot over here, factors in the abundance. So you can, on the left side, every individual species or strain that we are able to determine from our studies counts for 1. In this one, abundance modifies that. And in both cases, what you see, and this is David's passage. So when he's down there working on this project, he lives like his family. He eats what they eat, boa constrictors and piranha and tarantulas and grubs, carnivores, and they eat manioc and plantains as well. On the far right here, this is what his family's diversity looks like. So within a relatively short time, his diversity went from where we are, and this is labeled as healthy skin, but I would just argue that it's just Western skin, to what his family looked like. It didn't take very long for this to happen. And the same sort of pattern happened, although it essentially happened more quickly when you looked at the abundance, because some of the things that were very abundant, he picked up very quickly. This is just another way of transforming that data, but as you can see that as he started, he looked like us, then he picked up their abundance. And when he got back here, essentially as soon as he got back here, he went right back down to where they were. And there was something in that that we missed early on that I'm going to come to in a second. These pie charts really represent essentially if I were to swab your skin, and every species on there, I gave it as big a piece of pie as it had the amount of real estate in terms of your microbiome. This is kind of what we look like over here on the left. This is David before he left. And what we have is a handful of species, about 4 species on our skin, have almost all of the pie. And then there's one little piece of pie here that's crowded with a number of other species, but most of them. This one over here on the far right, that's what the Yanomami look like. As you can see, there's much more abundance, and more importantly, it's much more balanced. This is David at his most abundant when living in the village, and everything that's outlined in red is something that he picked up while he was there, and then again lost. And this is, again, I put this together for a technical audience. There's a lot of names in here. But here was the critical piece of insight. If I were to run a swab on your skin or my skin, 90% of the DNA I'd collect would be human, and 10% would be microbial. And I'm not going to get an awful lot to start off with anyway. But if I run that swab on the skin of the Yanomami, only 30% is human and 70% is microbial. And I get more DNA to start off with. They have a much thicker, richer layer of microbes on their skin than any of us do. And most of the diversity is in that layer. And the big window into this, this is actually fun. There's a guy up in Guelph at the university there outside of Toronto who studied zoo animals. And he looked at their skin microbiome. And if you look at the diversity on the Yanomami microbiome, it looks very much like the diversity on zoo animals. But if you look at modern humans here, what you can see is we're way below everything. We have lost most of this diversity. I'm going to go past this. This just shows, well, very briefly, these are metabolic pathways. Remember I said we not just lost the taxa, the specific species, but we lost their contribution. So on the left, each one of these bubbles here is a metabolic pathway, and it is the degree to which it is represented on the skin. As you can see, we do a couple things very well, but we've lost a lot of the stuff here on the diversity. And if you look over here on the right, the Yanomami have a much more balanced picture and many more bubbles than we do. And with that came a corresponding loss in function. I'll go past this if you guys have any questions. This is kind of interesting, though, if we look at the pathways that we lost and what we call them. So each one of these lines here is a pathway. And this is the Western, it's the Human Microbiome Project data. And you can see that looking at us today, we do a couple of things really well, but there's a whole lot of stuff we don't do at all. And if you look at the Yanomami, there's a much more balanced representation. And David's progress here, he picks these things up as he's there. But what's even more interesting is if you look at what we name the pathways that we are missing, things like toluene and xylene degradation, biphenyl degradation, fluorobenzoate, these are not things they've ever been exposed to. We actually don't know what these pathways are for. We named them for things that we could identify that they do in a lab, but that is not what their function is in terms of our physiology. Again, representing the fact that we just don't know what a healthy human looks like. And this just compares what it looks like. This is from their skin from their chest, and this is from the river water. And what you see is, although the abundance changes, there's a huge overlap. And that's because they're riparian and they go in the river once or twice a day. And if you look at the microbiome of their stool, with their shit, you'll see that their shit and the river water and their skin have a tremendous amount of overlap, not so much with the soil. Again, illustrating the point that these connections are deep and long-lasting. They are connected to the world in which they live. For these people, human rights and land rights are exactly the same thing. They are indistinguishable at a biological level. And then we did some metabolomics. We looked at what metabolites they have on our skin that we don't have. And basically that they have molecules that they have on their skin that have never been found in human samples. 883 molecules they have in their skin that have been found in human samples but are never found on skin. 160. There are about 1,000 metabolites they have on their skin that are part of their skin that we don't have at all. And this is where it got pretty interesting. The woman here in the center in the Adidas shirt is David's mother, and on the way back, he brought her back. She wanted to visit some of her. She was married to David's father, who was an American anthropologist in the 70s and 80s. And the headman of the village, he was down there studying them for about 12 years. The headman of the village betrothed his sister, which is Yarama, to him, and his father married her and brought her back here to the US for 5 years and had 3 children with her. She did not like the way that we live. They are connected to their family and the environment. We live in boxes. And so on one of their trips back, she ran into the jungle, and David didn't see his mother for 20 years. He had last seen her when he was 5. He got a degree in biology, and then he mounted this expedition. Yarama wanted to come back one last time to see her other children and grandchildren, and apparently she missed pizza and French fries. On the way back, she took a shower. So remember, this is what I told you that our percent human reads is about 90% for us and about 30% for them. After she took a shower, what you can see is she lost a lot of these diversity. One shower with soap. Now remember, these people go in the river once or twice a day, but the difference was soap. And I think I have an even better example of this. This is behind the ear, which is one of the places that has a great deal of diversity. This is before the shower, and that is after the shower. And what we've learned here is that everything that we think that we know about the skin microbiome today is about the epidermal microbiome. It's the stuff that is engrafted into the skin. Let's go past this. Again, her back did not see this diversity, but again, she can't wash her own back with soap. And again, you can see on this slide all the diversity that was lost with a single exposure to soap. So what we used to have is a layer on top of our epidermal microbiome. It was much richer and much more diverse and developed and made all of these important biomolecules, vitamin A, D, E, K, ubiquinones, phytosterols. And this layer sat right on our skin. And every animal that has ever lived and every human that ever lived until we had soap had it on it. And our skin gets between 15 and 50% of the cardiac output at a given point in time. And these molecules can move through the skin barrier and are absorbed systemically. And when we use soap, remember I talked about we pull blocks out of the tower? When we invented soap and decided to use that, the first time we did it, we washed that away. And none of us have that anymore. And it's not so much that we're missing the microbes, because we can't put the microbes back, but we're missing all of the metabolites they made for us. And when you take these things away, they have systemic implications as well as implications in terms of our skin. Because they're absorbed, there's a number of different ways they can be absorbed. They can absorb, directly through the cells. They can be absorbed through the skin barrier, and they can also be absorbed through things like follicles. But none of us have it anymore. We have a thin shadow of what was our evolutionary legacy. Okay. Well, here's what we learned, that our health and fitness depends on our relationship to the microbiome. And inadvertently, we've made decisions being unaware of their implications that have cut those relationships. And those have implications for our health. And this may be one of the reasons that we have such a burden of inflammatory disease, which is that contributed to our resilience in response to stress. And when you have lots of resilience, the tower can stand in response to stress. But when you lose the resilience, even a small stress can cause the tower to topple and fall. All right, we're almost done here. Look, everything that the way we see the world is a function of our perspective, and our perspective is determined by our identity and the way we see the world. I'm going to bring this back to Descartes, who told us that because we think, we're different and the rules don't apply. But the rules do apply. We are not apart from nature, we are a part of it. And it is our identity and our context that give us our perspective, and it's through this perspective that we see everything. And until we change this perspective, we're going to continue to make the same mistakes over and over again. And I'll illustrate this for you, and I can't see you, so you'll have to take a word for it. I'm going to give you all a word. I want you to call forth an image in your head. You don't have to say what it is. All right, the word, nature. Give you a second. All right, I'm going to assume that all of you have an image. I'm also going to assume that none of you have any people in that image. I've been doing this for a long time. Nobody ever has any people in that image. We took ourselves out of context. And then we solve these problems out of context. And then context reasserts itself, and we're constantly fixing the last thing that we broke until we put ourselves back in nature. The same way that the Yanomami are, the same way that was our evolutionary legacy. We are going to continue to make the same mistake. Nature has literally sent all of us to our room to consider our past behavior. Let's hope that we make better decisions. Thank you.
Okay, you guys, now that you're all totally depressed. I've been listening to this, parts of this information from Larry for as long as I've known him. And it influenced my behavior. I won't go any further than that. Anybody? Yeah, questions? Other than amazing. You know what, Larry, I'd like to ask you to describe one thing about how you test ingredients for their effect on the microbiome, or generally that you do. So
right now you're seeing people out there talking about products being biome friendly or not biome friendly. You know, I'm very skeptical about this for the following reason. The first thing is that our microbiome, this is the weeds of the weeds. With everything that we have done with it, if something was going to be sensitive to any of the chemistry, it is long gone. Your skin microbiome is basically locked in right sometime in adolescence. And it is next to impossible to either remove an existing species or strain as it is to add something to it. This is called colonization resistance. Now, if you think of the pie chart, you can alter the relative abundance and composition. So for example, if you apply something which is a prebiotic and selectively gives an advantage to some species or strains, then you can see their abundance grow. Or if you use a selective antibiotic, which selectively disadvantages some, you can see that one shrink, and it will, and it's an ecosystem, so something will take that niche. And in those shifts of abundance, you can create what's called a dysbiosis that might either trigger inflammation or perhaps even resolution of inflammation. We don't in fact know. But the idea that we can look at how an ingredient might affect the microbiome and do that in a way that gives useful, actionable information, that is way out in the future, and it may not even have any clinical correlation at all. What we have decided to do with Symbiome is recognizing this. We're just looking to replace their metabolic contribution. So we study the metabolomics of what the ancestral human microbiome looked like. We look at the metabolic pathways within this complex ecosystem. We have isolates from most of these organisms. We get them, we grow them up, we get them to express the gene products that they made when they lived on our skin. We are not putting any live bacteria in any of our products. We are only putting the metabolic products from it. And then we run clinical trials to see whether or not we're getting the outcomes we're looking for.
We have a couple of questions. One is the implication that we should only shower with water and no other products, which I'm sure you've heard before.
No, it's, well, look, I think that we're all doing science experiments on our skin, some more than others. The average American woman apparently applies over 100 individual ingredients, many of them xenobiotics, which means they didn't exist anywhere in nature until we made them in a lab, every single day on their skin. And when they don't deliver on their promise, we apply more of them. And every time we do that, we set up the possibility for some sensitivity or adverse response. And right now, over 70% of women, and most men too, think they have sensitive skin. And why wouldn't they? But just not showering or not using soap, look, we're not going to get back our ancestral microbiome. And if we stopped using soap, this third layer of the microbiome, which by the way, we're writing this manuscript up for publication. I'm going to see if I can get it called Schmutz, in tribute to my grandmother, who told me many times I should wash that schmutz off my face. We will not get back ancestral schmutz. We would get back the schmutz from living in a built environment. And I'm not sure that that would make any significant contribution to our health. We are who we are. That is, that is the case. The question right now is, how do we get back some of our resilience? And based on what we know today, the only alternative to this is replace the lost function, because we can't replace the lost microbes.
So the next question sort of leads from what you just said. And it's from Dale Davis. It says, loss of diversity making us sick. Does this require more specific species grown in a lab? Is this viable?
No, it's not. There are a number of, the microbiome science falls into sort of two groups. There's the consumer side and there's the therapeutic side. On the therapeutic side, there was this, oh, well, we need to increase diversity. How do we increase diversity? And the overwhelming conclusion right now is we can't. It's certainly not on the skin. There may be ways to do it within the gut, but still it's looking very much, literally you can run alcohol across your skin. And a week later, it's going to look just like it did before. It will grow back to where it was before. The roots are deep within the biofilm of the skin. And David had this thick layer of ancestral microbiota on his skin after being there for several weeks. And as soon as he came home and took a shower, he looked exactly like he did before. If there was any way to re-engraft these ancestral microbes, you would think it would be to wear that skin for a couple of weeks. And it didn't work. So I believe that what happens with us is by the time we're adolescents, our microbiome is essentially locked in. So things that are grown in labs may produce metabolic products that are useful. That's something that we are working actively on. But, and there's a difference between functional engraftment and actual engraftment. So if you take a probiotic, and by the way, you can do this in terms of a pill, or you can do this in terms of fermented foods. They may never become engrafted. In other words, when you stop taking it, they will soon be gone, but they may be functional, and you can get the functional contribution from them.
Have you seen the work that was done at Berkeley where they measured the shared microbiome from people walking past each other in a contained space? I just have this visual memory of, I saw the paper a long time ago. Do you know what I'm talking about?
I do. In fact, what we just discussed internally, there's a paper about people who cohabitate with each other.
Right. Yeah, that was another one I saw.
And it turns out what happens is cohabitating changes the exposures that you have. So the relative abundance of specific species tends to start, although never really completely, look a little bit like each other. But there is no transfer of species or even strains.
Really?
Yeah, it looked like it before, but now remember what I told you about our foundational methods. And by the way, I do have a really hard stop after this. I have to talk to beauty editors in 9 minutes, but this is an important point worth mentioning. Remember, if I swab your skin, I don't get very much bacterial DNA. So we're still doing what's called 16S. We're still looking into the barcode, and it has all of those things. It does not give you strain level data. In most cases, all you get is genus level data. It's very crude. The alternative is what's called shotgun sequencing, and I sequence all of the DNA that comes off. And remember, 90% is going to be human, and I'm going to throw that away. But that gives me insight not just into the ribosome, but into all of the functional components there. And it gives you a much more in-depth picture, but it is much more expensive. And each sample can cost $500 or $600. You know, that's what we did with the Yanomami. We did shotgun sequencing. But it's impossible at this point to really make the determinations of, are there strain level transformations between people who live together? And it's possible, because if you have an existing ecosystem and you introduce something that is the same species but a different strain, there's a chance that can get fit into the ecosystem. But if you introduce an entirely new species, there is no niche for it. There is no way for it to shoehorn in.
Larry, if you need to go, thank you. And go ahead and
I got 5 minutes if you guys want.
Well, this is a great comment here from Ellen Para, who says, do you think post-pandemic we'll find more biodiversity? Brink of extinction can yield a boom of biodiversity. I don't know that it yields a boom of biodiversity. I know that stress causes plants to grow more right before they die.
It's an interesting question. I don't know. There's a pattern in biology that after a crisis, like a forest fire, there's regrowth, and in regrowth, typically you see, you know, a boom in diversity compared to what it was before. But you know, anytime an ecosystem goes through a period where it's fully occupied to there's some sort of crisis and opens up a bunch of niches, there is a sequence of recolonization. There are pioneer species that come in and they prepare the ecosystem for the next one and the next one, but they have to happen in a certain sequence. So look, I'm just hoping that we learn something, and that what we don't learn is, see how clever we are. We made a vaccine so quickly, we don't have to worry about this anymore. Because if we don't learn something, I think the next lesson will be harsher. Mm-hmm. And you may not be able to tell, but I am a pathologically optimistic cynic.
Okay, well, then with that, Larry, I think we will let you go and at least take a breather and a drink of water before you talk to beauty editors, because they can be a lot rougher than we are.
Yeah, that's true. I enjoyed this. I hope you guys did as well. If you have any more questions,
absolutely,
feel free to reach out.
It was awesome.
Yes, it's wonderful, and I'm so glad to be able to share you with all of these other people. And this will be on YouTube. Is that's okay with you, Larry?
Yeah.
Okay. So thank you all again, and make sure you keep an eye out. I think we're getting a little better about getting our newsletter out and giving people notice. We're also all over Instagram for NOHBA. And with that, thanks everybody. And we will sign off and hope you have a wonderful day in the cold of the United States. Stay warm, stay safe, stay healthy.